following investigation is a forensic audit of the human biological record. As a journalist and digital content creator, my objective is to cross-examine the raw data provided by mainstream scientific institutions and compare it against the historical and geographic record. The information presented here is sourced from peer-reviewed publications, including Nature, Science, and the British Medical Journal. However, this audit is independent of interpretive summaries often attached to these findings by academic and governmental bodies. We are bypassing the narrative to examine the primary genetic signatures that define the human tree. This report is provided for educational and investigative purposes. Our goal is to address the onomastic displacement and phylogenetic conflations that have, over time, obscured the original identities and migrations of the ancient world. Transparency and accuracy are the pillars of this work. Every citation is provided in full, and viewers are encouraged to perform their own due diligence by auditing the sources themselves. We are entering a phase of discovery where the biological ledger is being read with a new level of precision. This is not just a look at the past, it is a restoration of the record. Proceed with an objective mind. The audit begins now.
His work and the impact of his work very nicely. I won't read you the title, you can read it, and ask Dr. Hammer to step up here and give his talk.
Thank you. Well, it's really a pleasure to be here and to have the opportunity to address our campus and our community on this topic of human origins using DNA to trace our origins. I've chosen to highlight three different themes in my talk tonight. The first one is our place in nature, and the second one is the evolution of the traits that make us human, trying to track both of those, not only genealogically, but in history as well. These are issues that Darwin addressed directly. And then the third theme will be something that's related, but something that Darwin didn't address, because this is something that has been debated most intensely in the last couple of decades. It's a topic that deals with the origin, how our species originated. The modern human species. And in order to do the work that I do and to address this issue in general, genetics isn't enough. It can't stand up on its own. We need different disciplines to work on this together to give us the full picture. We use paleontologists are involved in this topic as well. They study fossils to infer the morphological changes that have occurred over evolutionary time. Archaeologists who study material culture, stones, stone tool kits, to trace the history of our species, to infer the behavioral changes that have occurred through time. And then, of course, genetics, the area that I'm involved in, using both protein work and DNA work to infer our genealogical history. And one of the points I'm going to make in the talk tonight is that the genealogical history for different parts of our genome can tell us different things about our history. And that's one of the challenges that we face in trying to put together a story, a full story about our history. The history of the field of genetics actually goes back to, as applied to anthropology, goes back to the early 1900s in work that was done at the protein level and up through the 1960s. And I'll talk a little bit about some of the work that's been done with protein, and then the transition into DNA work in the 70s through the 90s, and studying individual genes and the sequences of those genes, and then in the last few years having entire genomes sequenced that we have at our disposal to make inferences about. Now I'm going to try and limit the technical information as much as possible, which is a challenge in itself, but I will probably convey a lot of the ideas to you through this drawing, these tree drawings. So on the left you can see a tree that may be a tree that was developed by anatomists, comparing the anatomy of humans and great apes and inferring their relationships. That's a species tree. In the middle, I'm trying to convey to you this timeline of fossils as they've been discovered and dated, creating paleontologists will infer the taxonomic status of a particular fossil by comparing it with other fossils, its time depth in the fossil record, and make inferences that almost look like a tree. You can see these branches that I've shadowed underneath these timelines of fossils. And then on the right is a tree that is a gene tree. It's not a species tree, but it's a tree that reflects the genealogical history of a particular region of the genome. And the time frame for the genealogical history can come from the number of mutations that we see in the DNA, using some kind of a molecular clock approach to infer the time depth of the tree. Now, let's turn to what Darwin said about human evolution. We can summarize it in one sentence. In his Origin of Species from 1859, light will be thrown on the origin of man and his history. That's all he said in the Origin of Species. And if we look a little further in the literature, we find that he wrote to his friend Alfred Russel Wallace a couple of years before he published The Origin of Species, 1957, when asked would he address this issue of human evolution. And what he said was, "I think I shall avoid the whole subject, as so surrounded with prejudices, though I fully admit that it is the highest and most interesting problem for the naturalist." But his ally and bulldog, Thomas H. Huxley, just 4 years after the publication of The Origin of Species, published his own book called Evidence as to Man's Place in Nature. And he wasn't shy about tackling this issue about human origins and applying evolutionary theory to human origins. What he did was he did an extensive comparative analysis of human and ape anatomy, embryology, and ethology, behavior, as well as looked at the very, very few fossils that were known at the time, and concluded that humans have a close evolutionary relationship with the great apes, in particular the African apes.
History is often a collection of agreed upon lies, but the biological ledger is written in a language that cannot be falsified. Your Y chromosome is a non-recombinative data security protocol. Unlike other parts of your DNA, it does not mix or shuffle. It does not succumb to merging of distinct lines into one. It is a read-only document passed from father to son in an unbroken chain of custody. To change your history, they have to change your blood, and they cannot. But to keep you from reading this ledger, they had to convince you that you were a revised edition of a primate. They sold you a 98% similarity myth to anchor you to the earth, while the raw code proves you are a distinct, high-intelligence protocol. The audit begins with the very tools they use to muffle the record. Pioneered by Luigi Luca Cavalli Schwartzfa, Stanford, and Michael Hammer, Arizona, to track paternal migration through neutral genetic markers. It was initiated to track human migration through neutral genetic markers because the Y chromosome is passed only from father to son. It provided the first scientific way to track paternal lineages without the interference of maternal mixing. This research was quickly utilized to anchor the out-of-Africa hypothesis, using the genetic stability of foundational lineages to falsify a claim they were archaic precursors. During this era, the mainstream narrative pushed the claim that humans are 98% plus genetically similar to chimpanzees. Our forensic audit note this figure was achieved by selective sequence alignment, purposely ignoring indels, also known as insertions and deletions, and structural variations, while Hammer's error pushed near identity. Auditors like Roy Britten of 2002 proved that when you include the missing chapters of the ledger, the similarity collapses to 95% or lower. A 2 to 5% difference in a 3 billion letter code represents 60 million distinct data entries, not a match, but different operating systems. We will also note that Dr. Jeffrey Tompkins, one of the most prominent internal auditors of the human primate connection, his findings, he discovered that 98% figure only existed because researchers threw out data that didn't match. When he audited the entire genome, including the unalignable portions, he found the similarity was likely between 84 to 88%. The logic behind this He argues that the human genome contains orphan genes, taxonomically restricted genes that have no counterparts in apes, proving distinct foundational record. Some of the epicenter where this research took place were at Stanford University and the University of Arizona, where scientists began collecting global samples to identify the root of the human tree. The first management system used haplotypes, clusters of genetic markers inherited together, leading to the discovery of the Y-allele polymorphism, also shortened YAP, insertion as a defining family seal. Remember this seal, we're going to come back to this later. Before 2002, there was no unified language. Some labs used Roman numerals grouped 1 through 10, others used letter-number combinations that didn't align. This fragmented system created a scientific fog. Researchers could talk about the same paternal seed using different numbers, making it impossible for the public to identify the simultaneous stability of the paternal lineages. It kept the master seed hidden in a forest of conflicting labels. In the early 21st century, the system shifted to the single nucleotide polymorphism, also known as SNP or the acronym SNP. This testing allowed for the identification of the exact paternal seal in the code, leading to the alphabetized haplogroup systems A through T, created by the Y-chromosome Consortium, YCC, around 2002. They published the birth certificate of the modern tree, and genome research officially returned the Roman numeral system. This system is now managed by the International Society of Genetic Genealogy, ISOGG. While this unified the data, it also laid the groundwork for categorical overlaps, where distinct tribal seeds were grouped into broad haplogroups to hide the distinct peoples of a global region. This created the phylogenetic conflations and the onomastic displacements we see today. Companies like Family Tree DNA and 23andMe brought these tools to the public but introduced further categorical overlap, rebranding tribal DNA with generic geographic names like Middle Eastern to muffle the specific forensic record. A SNP is a permanent biological paternal seal, a single letter change in the genetic code. A son cannot evolve a primary SNP his father lacked. He can only inherit the seal or carry a new one forward. Joblin and Tyler Smith, 2003, the human Y chromosome. This study confirms that the Y chromosome escapes the shuffling of recombination, functioning as a molecular barcode for specific lineages.
We decided to pursue an approach where we would do this in a computational way and screen larger regions of the genome looking for introgressed fragments. And the theory behind this is simple in that if we have a model of divergence where two groups diverge in the deep past, they stay separate into separate lineages, such as one leading to archaics, one leading to moderns. And there's a short period of recent interbreeding where some of the DNA is now exchanged. What the genetic pattern might look like in that kind of model is, for example, if we have these two bars representing chromosomes in modern humans and these in an archaic form. In the first generation, you have a hybrid where each chromosome, one coming from a modern parent, one coming from an archaic parent, form distinct full chromosomes. But in the process of recombination and many generations later, these orange chunks, these archaic chunks, get broken down into smaller and smaller pieces. And so our plan was to look for these small little chunks of DNA by the signatures that were specific to the interbreeding process, which I don't have time to go into. But given that we didn't have a reference sequence for fossil DNA from Africa, we decided to use genomics and this computational approach to scour the genome for these kind of introgressed fragments. So we applied our approach to a data set of sequences we gathered from a bunch of different African populations. We found evidence for several introgressive haplotypes in different parts of Africa, but the frequencies of the introgressive haplotypes were highest in the pygmy populations from Central Africa. Using extensive computer simulations, we first showed that the data were not consistent with a model of no admixture at all. So we rejected the null model of no admixture. And using a likelihood approach, we were able to make inferences about the model that involved introgression. In particular, contemporary African populations seem to contain about 2% contribution of genetic material from an archaic form that introgressed approximately 35 to 40,000 years ago from a group that split perhaps as long ago as 700,000 years. And given the distribution of these different fragments, we suggested that the interbreeding may have been centered somewhere in Central Africa. Now, I just want to digress for 1 second here about a very interesting story that came up this year that involved the Y chromosome, and that is the discovery of a very rare and ancient Y chromosome that didn't fit into the known picture of Y chromosome diversity. Many, many years of research on the Y chromosome told us it was very much like mitochondrial DNA. All the Y chromosome variation today trace back to a very recent single ancestor that lived in Africa about 100 to 140,000 years ago. This was the picture that we had at the time, and it made a lot of sense because the most divergent lineages on the Y tree were found in hunter-gatherer populations like the Khoisan and the Pygmies, which was very similar to autosomal patterns. Well, this particular lineage was discovered in an African-American man from South Carolina who happened to submit this DNA to the National Geographic Genographic Project, which sees thousands and thousands of samples, was very unusual, did not fit on the tree, and when we dated it, we found that the TMRCA was pushed back to over 300,000 years ago. And interestingly, when we did extensive database searching to see if we could find where else in the world a chromosome like this might exist, we were fortunate to find in Africa, out of thousands and thousands of chromosomes that we searched through, there were 11 that seemed to be very closely related to this chromosome. And it turns out all 11 come from one little tiny region of Western Cameroon from a farming group called the Embo. Now, I just highlight that because the Embo live very, very close to this site that Christopher was talking about, the Iwo-Lero site, where there's intermediate or mosaic forms, sort of intermediate between archaic and modern groups. Well, so just to update where we stand today, now we have to add another admixture process in Africa, along with the two outside of Africa. And I just wanted to end by saying something about what kinds of models are supported by the genetic evidence. Certainly we can no longer believe that the replacement model is correct. Even after 25 years of dominating as a paradigm, it seems to have fallen. And the new models that seem to be relevant are models that were also proposed many years ago based on fossil data. I think that this is probably the most reasonable model. It's a replacement model with hybridization, but I'll skip over that as there doesn't seem to be a single location for transition in Africa. There's no single Garden of Eden. There's transitional forms that are spread very vastly across the continent. Therefore, there's certainly the opportunity for gene flow among these morphologically diverged groups over a long period of time. And I really like the idea, Chris already described it, where you have the multi-regional model now restricted to Africa, so at a different geographic scale, but these traits that make us anatomically modern could have been flowing along this trellis network of gene flow among these morphologically diverged groups for quite some time until the anatomical form was assembled in a single package somewhere in Africa. And I really like the idea. And I think before we'll really fully be able to address this from a genetic perspective, we need to find the genes that encode these anatomically modern traits and look at their evolutionary histories. So I'll leave it at the top. And you can see how recent in time our species, Homo sapiens, emerges in the scheme of hominid evolution. So we can say, by looking at the fossil record, that we no longer would consider Darwin to be right in this count. Hominid origins are not equivalent to human origins. And we must think now of the early evolution of our specialness being one of being upright, where the larger brains came later. Most of us, I think, would say that part of what makes us special are the cognitive abilities that must relate somehow to our larger brains. But we're not quite there yet, because this slide is showing you sort of the encephalization quotient, which is a way of giving you the relative brain size to body weight on the y-axis and the time scale on the x-axis. You can see that if we compare ourselves to chimps, we have a larger brain relative to body weight at the top, and then the various fossil species that I talked about earlier in the middle. And you can see that the so-called bipedal apes have a slight increased brain size over that in chimps. The intelligent omnivores, those early members of the genus Homo, had a slightly larger brain size again, but neither reaches the brain sizes that we have at the top. So in order to get to the characteristics that really make us human, we have to go on to the next stage of evolution and talk about the transition to anatomically modern forms. Also, the toolkits of the various species that I'm talking about. It turns out that all of the species I've discussed have been using basically the same set of tools. In about 2 million and a half years ago, the first stone tools are found in the archaeological record, known as Oldowan tools, which eventually, about 1.7 million years ago, become a slightly more complex grade of tools called Ashwellian tools. Here you have Ashwellian toolkits, and you can see basically what they look like. It's hard to tell a hammer from a crescent wrench. So we're not there yet archaeologically, which is a reflection of where we are cognitively at some level. So in order to get into that and understand how we made the final transition to the Homo sapiens, the species that we are, I would like to bring up theme 3 of my talk tonight. How did we make the transition to modern form? Homo sapiens shown up again on the upper left, and now I've got some different players here, all in the genus Homo, and you can see Homo.
We must correct the primitive glitch. Can use stone tools out of frontier necessity, not biological primitivity. The Adamic or Stone era was a survival phase of a high intelligence population that already possessed the source code for civilization. Klaus Schmidt, 2000, Göbekli Tepe. The discovery of 20-ton T-shaped pillars with intricate carvings dating to the dawn of the Neolithic proves that high-level architecture existed before metal tools. This debunks the slow evolution of intelligence and confirms that the first sons of the ledger were already master builders. At the end of each chapter, we will do a quick forensic summary. Here's the one for chapter 1. We have established the origins of Y-DNA research and exposed the 98% primate myth as a forensic redaction of the data. We have tracked the shift from haplotypes to SNPs and identified the scientific babble used to muffle the paternal seeds. Most importantly, we have proven that paternal Y-DNA is an immutable ledger and that the Stone Age was not a time of primitive precursors, but a high intelligence frontier era where the original men began building the world.
Thoughts on a new project that I've been working on. It's actually in response to a challenge from an evolutionist, a well-known skeptic who jumps on me frequently. But basically it's people claiming that you cannot measure mutation rates today and use that to say how long ago mitochondria lived or how long ago Y-chromosome atom lived. You silly creationists, that's not the way it works. You have to use a phylogenetic rate, which is performed by assuming a common ancestry between two species, like say humans and chimpanzees, counting up the number of differences between say the Y chromosome or the mitochondrial genome, and then dividing by the time. And so if we separated 6.5 million years ago, allowing a little time for lineage sorting, maybe 7 million years ago between the last common ancestor, you take the number of differences, divide by 7 million, and you get a very slow mutation rate. It's called the phylogenetic rate. It's usually on the order of 10 to the negative 8, that's 1 out of 100 million, mutations per year, per nucleotide. So any given nucleotide, you'd expect it to mutate once every 100 million years. That's a slow rate. Well, I like to talk about mutation rates in terms of generations, as in the number of mutations themselves per generation, not the rate at which the letters change. And that gives you one mutation every 2,000 generations, 3,000 generations. I don't remember what the exact number is, but it's very slow rate of change, extremely slow rate of change. And that's the number an evolutionist is going to quote. That 10 to the negative 8 translates into, all humans picking up one mutation in common every few thousand years. Oh, wait a minute. That's different, isn't it? That's how fast the species as a whole changes. But the mitochondrial leave and the Y-chromosome atom argument isn't that. It's how fast do mutations build up in the population? That's a different question. It's not how fast the species change, it's how fast do the people in the species diverge from one another. There's no substitution in Y-chromosome atom. There's no substitution with this mitochondrial Eve hypothesis, as everybody's different. Not everybody is the same. It's how fast we're separating. And you can definitely use the genealogical mutation rate to estimate that. And when you do, you get a mitochondrial leave a few thousand years ago, not a few 100,000 years ago. You get a Y-chromosome atom only a few thousand years ago, not a few 100,000 years ago. If you use the evolutionary rate, yes, you get one that's very far back in time, and it's not a biblical date, and it actually would contradict the Bible if you do that. But in order to use that rate, you have to assume evolution is true. You have to assume humans and chimpanzees have a common ancestor, but we're arguing that humans and chimpanzees don't have a common ancestor. So if you're using the evolutionary number, you're actually using a circular argument, because the question is, do we have a common ancestor? You can't assume it and then use that in a number. So let's talk about the genealogical rate, the rate we can measure, and the reason why evolutionists will reject that number. There's two reasons why they'd say this number doesn't apply. One is natural selection. They would say, well, selection is going to remove most mutations. Okay, but in neutral theory, which is a very popular theory amongst evolutionists, most mutations are neutral. In fact, most mutations have to be neutral. It doesn't mean
To hide the ancient civilizations and their neighbors, the mainstream system had to stretch the timeline. They created the molecular clock, a theoretical device set not by observed biology, but by circular archaeological guesswork. They tell you it took 50,000 years for a house to move across a continent, but the ledger shows a much tighter, more explosive reality. The clock is set by forcing DNA rates to match contested archaeological dates. If the bone date is a guess, the DNA date is a fiction. This next information comes from Sheridan Bowman. Radiocarbon dating. In this British Museum publication, Bowman exposes the fundamental flaws in deep time dating. She details the reservoir effect, where organisms absorb old carbon from the environment, like deep ocean water or limestone, making a sample appear thousands of years older than it actually is. She also highlights atmospheric fluctuations, proving that the ratio of carbon-14 has not been constant throughout history. With this, we have discovered a glitch in the forensic data. Because scientists assumed a constant environment that didn't exist, they stretched the dates to fit the out of Africa evolutionary narrative. This allows them to rebrand the foundational basal groups as primitive ancestors rather than the contemporary master architects they actually were. Allow me to now explain the problem with fractionation. Bowman highlights that organisms do not absorb carbon isotopes in a uniform manner. This biological bias, known as fractionation, can make two organisms that died at the same exact time appear to have died centuries apart. Let's now observe the difference of pedigree versus phylogeny. Real-time observation, which is pedigree, shows mutations happening fast. Our next sourced information comes from Parsons et al., 1997, Nature Genetics. This landmark study found that the observed mutation rate in living families was 20 times higher than the theoretical rate used by mainstream science. When you apply the real speed of mutation, 100,000 years of history collapsed into roughly 5,000 years. Wow, that's a big gap. With that piece of information that we received, we now also have found another glitch. Mainstream science ignores this fast observed rate because it would bring the mitochondrial Eve and the Y chromosomal atom into the time frame of only 6,000 to 6,500 years ago. Wow, wow, wow. All right, this concludes chapter 2. It wasn't as long as chapter 1 because for those who aren't aware of what DNA is and haplogroups, I had to get them caught up. So the forensic summary for chapter 2 is we have dismantled the molecular clock by exposing the reservoir effect in the atmospheric fluctuations detailed by Sheridan Bowman. By applying the pedigree mutation rate, we have collapsed the false 100,000 year timeline into a forensic window that matches the biblical record. All right, we'll now be moving on to chapter 3. From here on out, we'll be picking up the heat. This forensic audit is going to get very good. And if you are here from my previous videos, let me know if you like the new structure, the new style, or do you prefer the old? Comment in the comment section. Don't forget to like, share, and subscribe. Thank you.